Good morning, good afternoon, and good evening, depending on where in the world you're joining us from today. My name is Glenn Sawyer, and it is my privilege to welcome you to this webinar hosted by Agena Bioscience. Over the course of this presentation, we will be discussing recent developments in pharmacogenomics and pharmacogenetics related to the identification and relevance of significant biomarkers, as well as review current guidelines and educational resources available to anyone interested in furthering their knowledge in the field of PGx. Throughout this presentation, you have the ability to ask questions by submitting them through the Q&A panel of Microsoft Teams. You should see the Q&A icon across the top panel of this webinar, and we will have a Q&A session at the end of the presentation. To that effect, it is my honor to welcome one of the foremost experts in pharmacogenomics, Dr. David Kisor. Dr. Kisor certainly needs no introduction, as his accomplishments and contributions to the field of pharmacogenomics are extensive. He's currently a professor of pharmaceutical sciences and pharmacogenomics and the director of pharmacogenomics at Manchester University in Fort Wayne, Indiana, where he is responsible for directing pharmacogenomics across programs in the College of Pharmacy, Natural and Health Sciences. Dr. Kisor is very engaged in pharmacogenomics education, having been involved with the American Association of Colleges of Pharmacy and Pharmacogenomics Special Interest Group in defining and refining competency statements in genetics and genomics for pharmacists, as well as leading the effort to provide the first master's program in PGx in the United States. In today's presentation, Dr. Kisor will review new drugs being introduced to the market that specifically mention biomarkers. He will discuss the various guidelines for clinical PGx and the resources available through CPIC and the FDA, as well as provide the clinical evidence and utility of pharmacogenomic panels. For the final portion of the presentation, I will briefly outline the current PGx offerings from Agena Bioscience, as well as provide details around our partnership with Manchester University and their PGx educational course offerings. Dave, the floor is yours.
Thank you, Glenn, for the kind introduction. As Glenn said, we'll be talking about biomarkers, guidelines, education, a number of different contexts related to pharmacogenomics. Specifically, I'd like to look at the number of new drugs over the last 20 years or so that have biomarkers mentioned in their FDA-approved labels. We'll talk about some resources of clinical pharmacogenomic guidelines. We'll describe evidence levels of the CPIC, Clinical Pharmacogenetics Implementation Consortium guidelines, and describe the U.S. Food and Drug Administration table of pharmacogenetic associations, along with their other table of pharmacogenomic biomarkers and drug label. We'll kind of put these together, compare and contrast these, and talk about a new effort coming from the Personalized Medicine Coalition. So if we look at this very informative image that is from a paper from Arbitrio et al. From 2021, it's really a nice setup for us for this discussion. It's really talking about biomarker discovery, and we're really looking for genetic variants that have a predictive value. We are likely familiar with a number of these from the pharmacokinetic standpoint. So a predictive biomarker that can, with interpretation, can affect how we treat patients. And does that patient have a certain biomarker form that results in a different response to medication as compared to other forms that other patients may have? And when we look at trying to identify these biomarkers, there's a number of different approaches. We can have very broad genotyping, genome-wide genotyping approaches, maybe targeted genotyping approaches, maybe using some panels. And here I'm not talking about clinical panels, but more along the research line of panels, where we have examples of up to maybe 1,200 genes that are looked at, maybe 4,500 plus variants that are looked at. And then there's the whole approach with whole exome or whole genome sequencing. And of course, there's pros and cons to each one of these. But the idea is to identify potential predictive biomarkers, and we're going to talk specifically about a couple of these and how we can go from identification of a variant and its function all the way through to clinical therapeutic recommendations and some ideas behind these. So from 2000 through 2020, we've seen an increase in the number of FDA-approved labels that have biomarkers mentioned. The highest number was in 2018, up around 23, and we're seeing more and more. So you can see the trend going upwards, which is interesting to think about from the standpoint of the information we have that can inform clinical decision making. But from a new drug application by a sponsor, by a manufacturer that has an associated biomarker, the idea is how can their product be used in combination with this biomarker to benefit patients more optimally, if you will. Certainly, there are a number of medications where we are approaching therapeutics. A large number of medications where we are approaching therapeutics without the biomarker information. With that biomarker information, how does that improve patient care? When we look across therapeutic areas, you can see the large majority, well, actually it's not a majority, right, 49.4%, but you can see the biggest component would be the oncology area. And these may be, prognostic type of markers or predictive markers, certainly, but we're also familiar with other therapeutic areas where biomarkers are employed. Just a few examples, certainly RYR1 and CACNA1S in anesthesiology and looking at inhalational agents and succinylcholine relative to risk of malignant hyperthermia as an example, or maybe in gastroenterology where CPIC relatively recently introduced the proton pump inhibitors guideline. Older guidelines from CPIC that have been updated in the cardiology area, which we'll talk more specifically about today, would be the statins guideline, and then also Clopidogrel. And you may be thinking, boy, every time I hear a talk about pharmacogenomics, Clopidogrel comes up. And it's come up for this talk mainly because there's a new update from the guideline that came out in early 2022. And another example could be in psychiatry, where we have the SSRIs, the selective serotonin reuptake inhibitors, the tricyclic antidepressants, and then neurology with anti-epileptic medications like oxcarbazepine, carbamazepine, and phenytoin. So these are therapeutic areas where we see biomarkers, and they are really across different therapeutic areas, which speaks to the fact that we need to have education of not just the primary care interaction clinicians with patients, but specialists also. Now that information is through 2020. In 2021, we've had 17 more drugs that have had new drug applications approved by the Food and Drug Administration that include biomarkers in their labels. And these drugs are new to the market. The biomarkers aren't necessarily new biomarkers. If you look at the far right column, you can see that many of these are markers we've had and have been using for a while. And they may be broad in a way or more narrow in a way. So HIV expression levels, example with HIV treatments or the like. I think more familiar to us are some of the drug metabolizing genes. So for instance, for belzutifan, you have UGT2B17 and CYP2C19. And as an example, when you look at the label there, it's related to the poor metabolizer status, really of both of those biomarkers that relate to increased exposure of the patient to the medication and an increased risk of adverse events, including anemia and other adverse events. So we have 17 more biomarkers again in 2021, which is the last full year of data that we have. Now, when we look at this on a percentage basis, since 2015, the last 7 years through 2021, we see well over 20%. In 2018, where we had the highest number, the 23 that I showed on an earlier slide, that's 42% of the medications that were approved by the FDA in that year that have biomarker or precision medicines, as the Personalized Medicine Coalition speaks of. And then again, in the different therapeutic areas, or broadly categorized here, the cancer area, again, approaching that earlier number of 49%, other diseases 16%, and then rare diseases 37%. So again, we're seeing more and more drugs entering the market, approved by FDA, the new drug applications, getting those drugs to the market with biomarkers associated. So let's go back now to the original image that I showed, this biomarker discovery slide. And what we're going to do is drill down here. We'll go through this image one more time, but we're going to really drill down to the biomarker translation part of this and look at the Clinical Pharmacogenetics Implementation Consortium Guidelines, PharmBar, the Pharmacogenomic Variation Consortium, and then PharmGKB, a broad database, really for which you can get to all of the CPIC guidelines and others, which I'll talk about. But as we identify biomarkers and we think about them from the standpoint of drug development, the identified biomarkers that might be looked at from a statistical standpoint, yes, there's a statistical relationship. They will make their way maybe into drug development early on in preclinical work, in vitro or in vivo preclinical, again, in animal models, looking at metabolic pathways when we're talking about pharmacokinetics, or transporter pathways, or drug targets that might be enzymes that are not drug metabolizing enzymes, and certainly other targets like receptors. So those could be the kinds of biomarkers that make their way into drug development. And if there's a certain hurdle or threshold that's met in preclinical, then the whole idea of biomarker testing needs to be considered throughout the other aspects of clinical drug development. That is, clinical studies in phase 1, 2, 3, and maybe beyond that. So with decision points that come along with what is discovered in those different phases. So let's go ahead and drill down and look now at really how a pathway is built from a variant through to a clinical recommendation. So if we have a biomarker that's identified and we look at it from its utility, in that, utilizing this test, a biomarker test, will it affect an outcome for a patient in a positive way? What is that utility of that marker? We see that the pharmacogenomics knowledge base lists more than 180, almost 190 clinical guideline annotations. So what does that mean? These guideline annotations come from groups that specifically focus on the question of biomarker testing, and what does that do for us with clinical decision support to better serve patients. So this is a screenshot from the clinical guideline annotations. You see the majority listed here on the far right upper corner, the CPIC guidelines in red, the Dutch and other groups. And let me fill in that other group information for you. There's the French National Network. There's the Canadian Pharmacogenomics Network for Drug Safety, looking specifically at drug safety. And there's some 2000s information that came from that group, which was very interesting relative to CYP2D6 and coding metabolism, and it's been controversial, but the point is that group looks specifically at safety. There's the American College of Rheumatology. So here we have really a therapeutic area group, a professional group that is not broadly looking at pharmacogenomics, but looking specifically at needs related to patients that their members would treat, let's say, or not necessarily their members, but individuals with that specialty would treat. So we've got these different kind of guidelines. There's a couple of others that are listed. This is the Fibrosis Foundation and Australian and New Zealand consensus guidelines, among others. So this is a really nice clearinghouse, if you will, and portal to get to these guidelines. And if we look specifically at the CPIC guidelines, we see over the last 11 or 12 years now, accumulated up to 26 guidelines. We've had a number of updates, 20 genes and more than 65 medications. And we see these across, again, therapeutic areas. So you've got cardiovascular, you've got infectious diseases, you've got rheumatology, you've got metabolic diseases, and so forth and so on, among others. And we're going to look at two specific examples here. And the reason we're doing that is these were examples that help us look at the path from a variant all the way through to a recommendation, but they were recently updated. And so there's some new information that we can talk about and put this in a broader context. I think that, you know, we oftentimes will see emerging drug gene pairs or drug biomarker pairs, and the data is just not at the level that will result in a clinical guideline being developed at a given point in time. But we see new guidelines coming. For instance, at the bottom of this slide in the lower right, you can see the G6PD resburicase original guidelines is going to be expanded with additional medications. CYP2C19 and SSRIs are going to be updated. CYP2B6, a new guideline relative to methadone, is coming. So we've got, and another big one is beta blockers and CYP2B6, ADRB1, ADRB2, GRK4, and GRK4 and GRK5. So there's a lot coming, but they weren't there before because the data wasn't there, right? And even within guidelines that have been published earlier, like the SLC01B1 and simvastatin guideline, you'll see when we talk about it a little bit later that it's expanded to the broader class of those statins, including atorvastatin, pravastatin, pitavastatin, and others, rosuvastatin, fluvastatin, and so forth. And then for CYP2C19, the expansion wasn't so much to a different drug here. We're talking about Clopidogrel, but the expansion was to a new indication, looking at the neurovascular indication. So I think we have these kind of in the pipeline, if you will, and different curation groups will look at things differently. Certainly CPIC looks at things differently than the Food and Drug Administration does, and we'll compare and contrast a little bit of that later on. So when we look at CPIC, there's really, I guess, two portals or two entryways for a drug to get into the system to be evaluated. One is, and we'll start with this, is where a gene is already subject to a CPIC guideline. So there's already information about the gene, its variants, and the activity and function of those variants, if you will. And so there we might be looking at alternatives or evidence that comes along that makes this guideline be used potentially in a different way. As I mentioned a minute ago, and as we'll get to, Clopidogrel now in the guideline from CPIC goes beyond the cardiovascular indication to the neurovascular indication. And then where does this fall as far as evidence? Is CPIC level A, B, you know, where is it that this new information or updated information takes us relative to levels of evidence and clinical decision support? This slide shows the different CPIC levels, at least through level B. There's C and D also, but for time's sake, I wanted to focus just on those that really are related to drug gene pairs that end up in guidelines. So CPIC level A is really related to genetic information that should be used to change prescribing of an effective drug. And this is based on the fact that the majority of preponderance of evidence really is in favor of changing prescribing. And that prescribing may be due to expectations of better efficacy or increased efficacy, or decreasing the risk of adverse events, or maybe both. And it's backed up by, you know, at least stronger or one moderate or strong action that can be recommended. Then we have AB, which is not a level that's been around from day one, but it's kind of in between A and B. We'll get to B in a second, but here there still needs to be review of the information, but it's likely that there's going to be actionable information coming forward. And this actionable means and you can find this actionability or informative or recommended or required testing, but actionable means that there's information in the package labeling that implies or is strictly there that relates to a dosage adjustment or maybe even a contraindication. As an example, with Clopidogrel, the box warning for poor metabolizers states that another medication should be considered. And then level B is genetic information could be used. Now, my background's in pharmacy, and I've got a background that really expands across different practice settings for pharmacists, from community pharmacy to hospital setting, and then certainly in academia and consulting. But I bring that up because we may be, as pharmacists, looking at issues that patients have, not from a therapeutic standpoint, but maybe, say, from an economic standpoint and their ability to pay for medication. As an example, years ago, we did a study with a group in West Central Ohio who wanted to include pharmacogenetic testing relative to their intervention patients in cardiovascular disease who were getting catheterized and then stents placed. And of course, the antiplatelet question came up. They didn't know anything about the pharmacogenomics. They didn't want to deal with it. So they chose to start patients on not a generically available Clopidogrel product, but at the time, a name brand Prasugrel or Ticagrelor product. And those were much more expensive at the time. Prasugrel has since gone generic, but the point is that they wanted to avoid the genetic testing issue because they didn't know how to deal with it. So that they put patients on medications that were very expensive, or relatively at that time. And the testing was done, and the information was used to change prescribing, not because of therapeutic issue, but because we could switch those patients to a generic Clopidogrel product, and they were able to afford it and maintain their adherence to their medication, which is, of course, very important. So that was Clinical Pharmacogenetics Implementation Consortium. We're going to connect these in a minute, but let's look at PharmGKB and their clinical annotation levels. Earlier, we saw 188 guideline annotations from PharmGKB, and I want to focus on level 1 here. And from the text in the right column under the description for level 1, I've taken an excerpt out where it says, or provide mapping from defined allele function to diplotypes and phenotypes, or drug label with varying specific prescribing guidance. So I want to walk through this mapping, if you will, from a variant to a clinical recommendation. We'll do this a couple of times. And the first example, as I mentioned now numerous times, Clopidogrel and CYP2C19. So PharmGKB 1A, CPIC level A. The first guideline on Clopidogrel and CYP2C19 came out in 2011. In 2013, it was updated. Both of those focused on the cardiovascular indication. And now in 2022, after, say, 2.5 years of effort, the update was published. And I'll talk more about that. But we start off with, on the left and the bottom of the slide, a haplotype. So there are 4 single nucleotide polymorphisms, or 4 variants that make up the haplotype of CYP2C19 star2, the star2 allele. And this is a no function form. Well, how did we get this no function form, right? And how do we understand that it's no function? Now, beyond that variant, we understand that a patient has information from their mother and their father. So we have the diplotype example here of a CYP2C19 star1, star2. And what does that translate to then in phenotype terms? Because it's the phenotypes that drive the recommendations in the clinical guidelines. And not just CPIC guidelines, but the Dutch guidelines and others. So here the phenotype is intermediate metabolizer. And then if you look at the guidelines, and we'll look at this in a couple of slides, it says to avoid standard dose of Clopidogrel, possible use alternative. So we went from a variant to a clinical therapeutic recommendation, but let's dig into this a little bit deeper. So where can we get this no function information from? One example is going to PharmBar and the Pharmacogene Variation Consortium. And you can see here for CYP2C19 star2, and if you look to the right where the arrow starts going to the no function at the top of the slide, near the top of the slide, you see the X under CPIC clinical function. That means no function. But okay, this is another database. It used to be called the CYP allele database, and it's mostly looking at the cytochrome P450s, but there's some other genes in there also. But this is not really where it comes from, right? This is just a database that's gotten information from elsewhere. Now, if we look at some of the background information of the CPIC guidelines, we have another way to get to more information. So in the upper left, we see the allele definition table. I've cut out the other variants that aren't pertinent to this context, but we see the STAR2 allele that has the blue rectangle around it. We see the 4 variants that make up that haplotype, and that is the definition as far as what variants make up that haplotype. And then we can move to the functionality table. And we see under this functionality table, the STAR2 has no function under column B in the middle panel. Now, again, that's a summary designation, right? No function. Where did that come from? That's what I'm really trying to dig down to. And if you go to the right column under findings, although it says optimal, these columns are populated with information. And when you see, for instance, in parentheses, the 8195181, the 2202765, those are 650, those are PubMed ID numbers. So it's going back to the primary literature, and that's where I wanted to get to, how we drill down with the individual variants based on the primary literature. And in this case, there were probably 4 or 5 or 6 studies that were definitive in showing that the STAR2 variant was a no-function form. And this was done with looking at markers of metabolism, S-methanoid as an example. And it's going to depend on the gene, it's going to depend on the markers that are used, but this is really where the data comes from. This is where the raw primary literature data translates to no function, then moves to the diplotype stage where you have the STAR1, STAR2 in the lower left, and you end up with the intermediate metabolizer phenotype. So that's the mapping from the primary literature related to that variant to the defined function, in this case no function for the STAR2, to the diplotype when you combine it with the STAR1 form, in this example for CYP2C19, and getting the intermediate metabolizer. And then the next step, of course, is going to the guidelines and looking at the interpretation of this information. So a little bit of redundancy here, though. This is the information that is taken directly from the guideline table 1 in the 2022 update. And this looks at the CYP2C19 intermediate metabolizers. So an individual carrying, excuse me, 1 normal function allele and 1 no function allele. That's the rectangle around the STAR1, STAR2. STAR1, STAR3 is also normal function, no function, or 1 increased function allele and 1 no function allele. And that's the STAR2, STAR17, and STAR3, STAR17 examples. Of course, we're looking at the STAR1, STAR2. All of those qualify, and there are others. I think there are some 37 variants listed in PharmBar for CYP2C19. Now, if you look at PharmBar, you'll see it goes all the way down to 39 at the bottom, but there's not 39 because there were a couple that were retired or changed somehow. So it's really, I think, around 37. At any rate, there's more than 35. We can say that. And when we look at those variants, there can be numerous combinations that will result in different phenotypes. And we'll talk a little bit more about the phenotypes in just a second. But this specific intermediate metabolizer phenotype then can be translated into a therapeutic recommendation. And this is from table 2 from the guideline. And this is noted in the cardiovascular indication. Here, avoid standard dose of clopidogrel if possible, use prasugrel, ticagrelor at standard dose if no contraindication. So what we start out with is this primary literature looking at a variant and looking at the clinical functionality, if you will, of that variant, combining it then with other variants to look at a diplotype, which translates to the phenotype, and then to a therapeutic recommendation. What we've done is gone from the variant primary literature all the way through to a therapeutic recommendation. Now, when we look at the CPIC guidelines for the 2022 update, we see a couple of extra phenotypes in here. One is a likely intermediate metabolizer, and the other is a likely poor metabolizer. These were not in the previous CPIC guidelines. And in fact, the previous CPIC guidelines used old terminology. Standardized terminology was presented in the standardized project from CPIC in 2016, where we changed excessive metabolizer to the normal metabolizer. But we've got two new designations now, likely intermediate and likely poor, and that's related to decreased function variants of CYP2C19. And you can see on the slide star 9, 10, 16, 19, 25, and 26. Those are relatively rare variants, but we haven't characterized the functionality completely, and therefore combining those decreased function with other variants results in likely designations as opposed to definite designations. The next slide, please. Also from the newest guideline in 2022, as I mentioned earlier, we've now expanded to the neurovascular indication. So if you think about the primary literature for a given variant and mapping it through to the star allele haplotype, through to the diplotypes, to the phenotypes and the therapeutic recommendations, now we have recommendations that are available for the neurovascular indications. And interestingly here, these are really specific to Clopidogrel as prasugrel and ticagrelor. Excuse me, prasugrel is contraindicated in patients with transischemic attacks or strokes. So they're not, that drug in particular is not useful in these indications. So again, we expanded the guideline here, not to more drugs, but here to a different indication. Okay, next slide, please. So if we now look at the other entry point of information into a CPIC review and guideline development, we can look at genes that are not yet subject to CPIC guidelines. And I want to point out here that pharmacogenic annotational 1A, 1B, 2A, 2B, or other reasons for moving a drug into this pathway can be considered. So here we'll look at this from the standpoint of pharmacogenic level 1A, and then where does that fall in the end to CPIC's level of evidence. Next slide, please. So this is a similar slide to what we saw earlier, where I took the excerpt out where it says, or provide mapping from defined allele function to diplotypes and phenotypes, or drug label bearing specific prescribing guidance. I've got two lists here, two maps, if you will. We see fluvastatin and CYP2C9, and I'm not going to talk about that in detail here because CYP2C9 was already a gene that was a subject of other guidelines, including warfarin, including phenytoin, and then most recently, the non-steroidal anti-inflammatory drugs. But what I'd like to focus on here is the new gene that's been included, the ABCG2, and related drug rosuvastatin. So as I did previously for Clopidogrel and CYP2C19, I want to show rosuvastatin ABCG2. So here we've got alleles, and we talk about these in this case from alleles. ABCG2 does not use the star nomenclature. So we have this RS2231142 variant where T replaces G, and that's related to decreased function. So that's the allele, and that's at that variant level. And if we go to the genotype, here we have a heterozygous, so G, the reference, and then T, the variant, and that relates to a phenotype of decreased function. And then the therapeutic recommendation here actually is to prescribe desired starting dose. So for a decreased function relative to rosuvastatin and ABCG2, we have a recommendation to continue with that standard dose, essentially starting dose, but then adjust based on other specific guidelines. And those are disease-specific guidelines, say for cholesterol, dyslipidemia, and that's the context. Next slide, please. So this is a similar slide to the Clopidogrel CYP2C19, where we started out with the allele definition. And here we only have one variant, again, the RS2231142, where T replaces G. And so T is the, I guess the, the actionable variant here. And when we go to the allele functionality table from CPIC, we see it's decreased function. And again, that's all well and good that that's been designated decreased function, but we really want to look at the primary literature, and you can see a number of papers that are listed by the PubMed IDs in the findings column. So again, we've got that primary literature, we have a designation for the variant then combined in the genotype or diplotype as noted here in the lower left, the genotype phenotype table, and you can see the different combinations. You can have the reference, which is a normal function, the reference invariance, which is decreased function, which is the example here, and they could also have the homozygous variant TT, which is poor function. So those are different examples of the genotypes for the patient, and then we move that along the line to the guidelines, where we have, again, a little bit redundant, but we see from the guideline table, and this is not the formal final version of this table. It's not been published in that manner yet, but we see decreased function, and you can see the C421C and A. Now here we have C and A, which is the complementary strand information. If we go back, we see we had G and T. So that's the difference from this slide to the previous slide. And then to the phenotype conversion, where it says prescribed desired starting dose and adjust doses of fluvastatin based on disease-specific guidelines and patient-specific populations. So these different progressions in these slides, going back to slide 30, then 31, and then 32, show you that path of the primary literature through the therapeutic recommendation. Okay, Glenn, please advance to slide 33. All right, so we've got information from the statins, which is new, and this guideline expanded the number of drugs and genes in this case. So where we expanded to a different indication in the CPIC Clopidogrel CYP2C19 guideline, here we expanded to 2 more genes beyond SLCO1B1, including ABCG2 and CYP2C9. The guideline has information related to SLCO1B1 for atorvastatin, fluvastatin, pitavastatin, pravastatin, rosuvastatin, additionally to simvastatin. ABCG2, our example here is related to rosuvastatin, and then the other example I mentioned, which I'm not going to go into greater detail because CYP2C9 has already been the subject of other guidelines, is fluvastatin. All of these are at high level evidence, and so this guideline now is greatly expanded and is going to aid in dosing recommendations, drug choice recommendations beyond just simvastatin. So again, we've got these CPIC guidelines. If you can go to slide 34, we've got these guidelines that have evidence behind them, and now we want to look at this relative to other information that people are developing panels or clinicians that are looking for information can utilize. So when we think about this, we think about 2 specific tables that have been offered by the Food and Drug Administration. So if you can go to the next slide, Glenn. We have the FDA tables. On the left, you see the table of pharmacogenomic biomarkers in drug labeling. This lists therapeutic products from drugs at FDA with information found in the label. So it's going to be related to different sections of the label. Is a biomarker found in the warnings and precautions? Is it found in the box warning? If there is a box warning, is it found in clinical pharmacology? Where is it that that biomarker exists? And that's important because it can help us think about what that biomarker can help us with. For instance, if a biomarker is in warnings and precautions, then it may be related to decreased efficacy or toxicity. If it's related to pharmacology, maybe that section of the label is talking about something that is informative from maybe more of a mechanistic standpoint. Now, the other more recent table that was provided by the FDA was in February 2020, I believe, is the table of pharmacogenetic associations. So in a way, the FDA said, okay, listen, we recognize there are these associations, and here's what we believe are associations that are significant in 3 different contexts. And I'll talk about the 3 different sections of the table in a minute. And then we'll try to look at these relative to guidelines and FDA tables. Next slide, please. So this table looks at the CPIC guidelines, FDA biomarker table, and the FDA associations table relative to the drugs that are mentioned in this presentation. So we see the drug genes column on the left, Clopidogrel, atorvastatin, the 2 genes that affect fluvastatin, SLC01B1 and CYP2C9, pitavastatin, pravastatin, and then the 2 genes that affect rosuvastatin, SLC01B1 and ABCG2. Of course, these are all just introduced a minute ago in the CPIC guidelines, right? So we know that they're present in the CPIC guidelines. Well, we then recognize that in the biomarker table from the FDA, we see Clopidogrel is noted, and it's also noted in the association table. And we're going to drill down a little bit further on the next slide with these designations, or at least these acknowledgements and these different resources. However, atorvastatin, fluvastatin, and pitavastatin, or I should say actually fluvastatin, pitavastatin, pravastatin are not seen in the FDA biomarker table nor in the FDA associations table. Atorvastatin is in the associations table from the FDA. And then finally, rosuvastatin is in all 3 tables, but interestingly enough, it's in the biomarker table and the association table from the FDA related to SLC01B1 and not ABCG2. So there's mixtures of what information is in these different resources. If we can go to the next slide, please. So in this slide, we drill down a little bit deeper into what information is in the slides, or in the resources, I should say. So again, CPIC for all of these medications are phenotype guided therapeutic recommendations. And again, we got to those therapeutic recommendations by starting with primary literature to look at different variants. When we started the process for Clopidogrel and used search terms CYP2C19 or cytochrome P452C19 and Clopidogrel, I think it yielded more than 690 publications. The last publication, of course, of the guideline was 2013, prior to this 2022 publication. And beyond that, there were some 360 plus papers that were reviewed. So that information is what led to the updated therapeutic recommendations in the cardiovascular and neurovascular indications. The biomarker table for Clopidogrel has the information in the boxed warning that's related to poor metabolizers, so 2C19 poor metabolizers, in the warnings and precautions part of the label, and in clinical pharmacology. Here, these are related to individuals who are at risk for major adverse cardiovascular events with Clopidogrel when they are really intermediate or poor metabolizer, but specifically poor metabolizer. And then in the FDA table, we see affected subgroups, and they talk about the poor metabolizers. Atorvastatin is in the FDA table, it's not in the biomarker table, and I should say FDA associations table. And then at the bottom, rosuvastatin again is in the FDA biomarker table and the association table related to SLC01B1, not ABCG2. Next slide, please. So I said earlier I would put some context to these different sections, or at least actually the FDA did, not myself. Section 1 is pharmacogenetic associations, really related to therapeutic management recommendations. Section 2 is related to the impact on safety or response, and Section 3 is really related to the ADME, the PK, absorption, distribution, metabolism, and excretion. And when we look at comparing now CPIC guidelines to FDA, we see that there are examples, or at least here an example of Clopidogrel, where the recommendations are essentially identical. CPIC says avoid Clopidogrel if possible, consider an alternative. FDA says consider use of another platelet P2Y12 inhibitor. So that's slide 39. Next slide, please, slide 40. Earlier this year in March, Pritchard and colleagues presented a really nice piece of work comparing FDA table of pharmacogenetic associations and CPIC guidelines. And it really compares contrasts information in the FDA resource and the CPIC resource. And there's really an appropriate presentation here from the standpoint of different approaches and different information, and you can get this from this new resource, and I'll talk about it in more detail. But here, there's different perspectives, right? A clinical guideline group versus the FDA. FDA looking strongly at safety of the public in a larger populations, if you will, from a safety standpoint. FDA having information from the manufacturers, these sponsors who submit a new drug application that isn't necessarily published and wouldn't be available to a guideline group. So there's different perspectives, different approaches, and different resources that are available for these tables that are developed. Next slide, please. So from this paper by Pritchard, we have three different categories here. CPIC guideline only, we have 20 drugs that fall in that category on the left. On the right, we have FDA table only, 35 drugs that fall in that category. And then in the center, we have those that fall on both the CPIC guidelines and the FDA tables. And this is related to section 1, where therapeutic recommendations are considered. So you can see that we have, these different categories, and the drug is going to move off of these individual lists as more data is gained, and we see the clinical pharmacogenetics implementation consortium information and FDA information matching up a little bit better. From the section 2 information, the FDA has 11 drugs on the right that are listed only by the FDA. Otherwise, CPIC and FDA have 7 that are considered in both of those resources. And finally, for section 3, the pharmacokinetics, we see 23 drugs that have pharmacokinetic implications relative to the associations table from the FDA, and 14 that are from both FDA and CPIC, and that's on slide 43. So we see this from an original publication recently, and we see that there's not complete concordance here, right? There's a lot of differences. So if we go to the next slide, number 44, when we have the original information from Pritchard et al.'s paper, we see that we have 20 CPIC guideline only, 39 CPIC guideline FDA, and then 67 from the FDA table. Now, the Personalized Medicine Coalition is going to be providing this information and updating it with a collaboration with our institution, Manchester. And so through Q1 22, we've seen 2 drugs move off the CPIC list to the CPIC guidelines and FDA list, where they're both represented, and 2 drugs move off the FDA table. So we see CPIC going from 20 down to 18. FDA went from 67 down to 65. However, vilazoxazine was added to move it back up to 66. And you can see the change log below, where fosphenytoin and phenytoin were moved from CPIC only to CPIC and FDA, and then atorvastatin and rosuvastatin from FDA to CPIC and FDA. You can see where viloxazine was added to the FDA table, and we also have a couple of other changes from PharmGK and CPIC that are noted here. So this is going to be updated and be provided so people can have an idea of where things are connecting between the clinical guidelines and the Food and Drug Administration. So underlying the use of pharmacogenetics, these resources, and going back to the primary literature, education is the key. Of course, I'm an educator and help out in other respects clinically, but we have programs at Manchester that we started first in the country in Master of Science and graduate certificate. And so we have some opportunities for greater education for individuals beyond the pharmacy education and those great residency programs that are out there. And we've also partnered with Agena to promote pharmacogenomics education. And you can see my contact information there. And in summary, biomarker evidence is needed, and it needs to be mapped right from the start all the way through therapeutic recommendations. These different resources are available that I talked about, the guidelines, the FDA tables, the evidence across resources can inform the development of testing panels. And then finally, education is the underlying key, if you will. So thank you for your time. And again, I apologize for the technical problems.
No, Dave, it's totally fine. I think it was, I love Microsoft, let me tell you that, but I think we were able to follow along. It was an amazing presentation. So thanks again for the in-depth and truly insightful presentation. It really is inspiring to see the span of PGx information and all the different resources available for laboratories that are interested in either entering PGx or expanding their current studies and offerings. And as a company, Agena is invested in furthering PGx offerings as well. So I'll be highlighting some of the tools and products available from Agena Bioscience over these next few short minutes. And it should be noted that the products I'll be presenting are labeled RUO for research use only. Our foundational PGx offering is the Veritos Core Panel. For many labs, this serves as a standard PGx offering as it targets 73 unique sequences within 20 principal genes that are implicated in drug metabolism. And much as we heard Dr. Kisor present on the importance of specific genes to determine drug associations and metabolizer status, Agena's high multiplexing technology enables cost-effective targeted testing of key PGx genes without needing fluorescent molecules, and with the ability for many labs to go from DNA to data in a single day. The panel's been designed to be resilient to lower quality DNA, which can be particularly important for PGx samples, which are typically buccal swabs arriving in the laboratory through various shipping methods, which can degrade the nucleic acids. Our PGx panel only requires as little as 5 nanograms of DNA, and the genes that make up the Veritos Core Panel can be seen here in the chart on the lower right-hand side of the screen. For PGx laboratories, CYP2D6, copy number variation, or CNV, it's a critical component of their offerings, as this provides crucial information regarding the metabolizer status of individual samples. Accurate analysis of CYP2D6 CNV is necessary because the gene, as we know, is highly polymorphic with multiple hybrid alleles, and these hybrids can lead to non-functional copies. Unfortunately, many of the competing technologies only look at 1 or 2 areas of the gene, typically exon 9 or intron 6, but this could lead to inaccurate detection of hybrid alleles, potentially leading to mischaracterization of metabolizer status. And the Veritos CYP2D6 CNV panel interrogates 7 different regions of the CYP2D6 gene with 23 unique assays, which allows for that accurate hybrid allele detection and CNV calling. And because of our technology, labs are able to run both genotyping and CNV calling within the same workflow on the same instrument at the same time, which is improving lab efficiencies and reagent utilization. The third key pre-designed panel from Agena Bioscience is our Veritos DPYD panel for the detection of 9 SNP genotypes that can be utilized for pharmacogenetic DPD activity testing prior to treatment with fluoropyrimidines. Of course, this is relevant because 10 to 40% of cancer patients treated with 5-fluorouracil develop severe life-threatening toxicities. These adverse drug reactions could potentially be avoided with PGx testing prior to treatment. And this panel, again, it can be run separately or on the same plate at the same time on the same instrument as the previously mentioned Veritos Core and Veritos CYP2D6 CNV panels. And for laboratories that would like to either supplement the existing off-the-shelf panels or create their own unique PGx content, we have an entire team dedicated to designing, developing, and performing verification. Our assaysbygina team has the expertise to work with you to create and deliver unique panels in all the application areas shown here on the screen, as well as many others. The team has the ability to design the assays, functionally test them to ensure robust performance, and deliver the panel directly to your laboratory. And many PGx laboratories utilize assays by Agena to supplement the core panels that we just reviewed. They supplement it with additional targets or other focus areas they're interested in interrogating. And of course, the Agena Bioscience panels run on the mass array system. This is an open platform that allows labs to focus on targeted sets of gene regions with known associations. This allows the targeting of tens to hundreds of genetic markers per sample in a single run, with a workflow that couples mass spectrometry with endpoint PCR, enabling the highly multiplexed reactions under universal cycling conditions to provide accurate, sensitive, and rapid genetic analysis. And the mass array system was specifically designed for efficient reagent utilization, using only the amount of reagent needed for the number of samples within your run, whether it be a partial or a full plate, providing efficiencies and cost savings to the lab by not having to wait to obtain enough samples for a full run or waste reagents when utilizing only partial plates. And many of the leading laboratories have found the mass array system to be cost-effective, rapid solution for their PGx testing. And as proponents for the power of PGx to transform precision medicine and improve healthcare, as Dave mentioned, Agena Bioscience is committed to continuing education and increased awareness, and we've been honored to partner with Manchester University in their efforts to create the PGx leaders of tomorrow and expand the skill sets of PGx laboratories of today. Manchester University offers the specialized programs, including the graduate certificate in PGx, as well as Dr. Kisor mentioned, the Master of Science in PGx degree. These programs provide students the opportunity to learn human genetics, bioinformatics, pharmacokinetics, and more as it all relates to pharmacogenomics. And this sets them up for success in this rapidly growing and increasingly important field. So to learn more about the educational opportunities through Manchester University, Dr. Kisor provided his contact information, or you can contact Agena Bioscience by reaching out to us directly through our website. And we're committed as an organization to the continued advancement of PGx, and we're continually evaluating opportunities to update our panels or create new content as the scientific evidence evolves, much as Dr. Kisor has reviewed here with changing information and scientific evidence. And to that end, should you wish to have an in-depth conversation with our scientific affairs team and discuss the potential of PGx within your laboratory, please reach out anytime through the email provided on the screen, and we look forward to hearing from you. With that, we do have a few minutes available for some Q&A. And again, you can submit your questions through the Q&A panel, and we've received several already. For any questions you may have that we don't get the opportunity or time to address here, please feel free to contact us directly through our website or through the emails provided, and we'll respond as quickly as possible. So one of the first questions that we have, Dr. Kisor, is which biomarker drug guidelines are used as the standard in the UK? Is it CPIC like it is in the US, or do they tend to use the Dutch Working Group or PharmGKB?
Yeah, so I'm getting some feedback. So yeah, it's a great point, right? There's the Dutch Pharmacogenetics Working Group, there's others. I don't know which are used primarily. There's the ubiquitous program in Europe. Likely the Dutch group guidelines are used. There's different practice standards in different regions of the world. I know that, for instance, the beta blocker information in the Dutch guidelines is very useful. Currently, CPIC does not have that guideline published yet, but it's coming. So it really does depend on different regions of the world and you know what their practice standards are. I hope that answers the question.
Thank you, Dr. Kisor. Next question is, what is your prediction on the creation and update of various CPIC articles based on new information, especially when some have not been updated in recent years? And some of the examples are CYP2D6, CYP2C19, and tricyclic antidepressants not being updated since 2016.
Yeah, CPIC has a schedule of work that's in progress, and they will be coming. There's just more information, and there's more work to be done. And CPIC is a voluntary organization, and so the information is going to be coming as soon as the guidelines are completed and reviewed to make sure that everything is as it should be before they're published. I wish we could have things, almost instantaneously, but that doesn't happen right now with really any of the guidelines. I mean, certainly the Dutch group too had published in 2011 and then more recently published. So there are these gaps. We recognize those gaps. CPIC is a very hard-working, great organization and working as fast as they can to get the information. But it's only going to happen as fast as information is provided in the primary literature, which allows for the kind of mapping that I talked about earlier.
Thank you, Dr. Kaiser. And in the interest of time, we have one more question that we'll ask, but what are some of the challenges you see primary care clinicians having in utilizing PGx in prescribing decisions?
Yeah, that's a great question. One is from the standpoint of clinicians that might receive different laboratory reports and how that information is presented. Certainly different testing panels have emerging gene or gene variants on their panels. And so they're not the same from lab to lab, or say from clinical decision support to clinical decision support. That can be confusing and can really, be a deterrent to using pharmacogenomic information. A recent qualitative study that we did, when we asked physicians, many of them just said, I'm going to ask the pharmacist. They're just not trained in this yet. It's not formally required in medical education. That doesn't mean that some schools aren't doing a great job at it. It's just that it's not ubiquitous. So there's a lot of challenges. Of course, there's the whole reimbursement payment. There's many different barriers, but all of that's getting better. Absolutely. I wish I had more time.
Agreed. And again, thank you, Dr. Kaiser, for always sharing your knowledge and advancing the field of pharmacogenomics, pharmacogenetics. And again, appreciate everyone for your time and attending this webinar. Any of the small hiccups are definitely Microsoft and my fault. Appreciate all your time and effort, Dr. Kaiser. And again, any questions, please reach out to us at any time directly. Have a great afternoon.